Now, when we talk about glycolysis, we're going to focus on glycolysis as a series of 5 reactions. There's actually more than that, but it's a little more than we actually need to get into. So we're going to look at 5 basic phases that are occurring to produce the ATP and to get us to the pyruvic acid. So you should have this diagram somewhere. I want you to find the full diagram. It should be in your packet somewhere. I'm not sure what page.
It is page 5.
Those of you that bought the packet, if you printed it, there's no page numbers. 21B.
Thank you, Dan.
Now, there's several things I want you to focus on as we're talking about this. I want you to think about the number of carbons. And I'll explain to you what all these symbols mean shortly. But I want you to pay attention to the number of carbons as we go through this whole process, not just glycolysis, but the whole series of reactions. I want you to pay attention to the oxidation reduction reactions. And of course, pay attention to the energy gains. Now, here's a key. A circle is a carbon atom. A diamond is a phosphate. And each of these dashed lines here represents the transition from one phase to the next. So why don't we go ahead and list the phases here. So up here, phase 1, this is called phosphorylation. What do you think that means? Say that again? It's involving a phosphate. We're actually adding a phosphate to a molecule. We'll get into the details in a minute. So the second phase then is what we call the priming phase. Three is the cleavage phase, because what we will see is that a 6-carbon molecule breaks down into 2 3-carbon molecules. Essentially, it's splitting in half. Step 4 is the oxidation phase, and step 5 is what we call the dephosphorylation. And what would that mean? We're losing phosphates. Any idea where those phosphates might be going? Remember, what's one of the products of glycolysis? ATP. And if we add a phosphate to an ADP, we get ATP. And that's what we'll see happening here. That dephosphorylation phase, we're going to see phosphates coming off and being added to ADP to make ATP. Okay? Any questions up to this point? Okay. Well, let's take a look at the phosphorylation phase here. Obviously, this is glucose here. Glucose, C6H12O6. I'm not drawing all the hydrogens in there, but they are attached, as are the oxygens. But glucose has 6 carbons, and so I'm just showing you that carbon backbone, 6 of them. So the first thing that happens is we have to use ATP. And we take a phosphate off of an ATP and we add it to that glucose molecule. There's our phosphate. It's been added to the glucose. And if we pull a phosphate off of ATP, what are we left with? ADP. Die. Which also means 2. And what we're left with is a molecule called glucose 6-phosphate. That 6 stands for the carbon that the phosphate is attached to. It's attached to the number 6 carbon. Now at this point, there's 2 options, or 3 options. Number 1, if conditions are right, the glucose 6-phosphate can continue the rest of the way through glycolysis. If extra ATP isn't needed, then that glucose 6-phosphate could get stored as glycogen. We'll talk about that probably on Wednesday. This process of going from glucose 6-phosphate to glycogen. We know that glycogen is a branched polysaccharide. It's a bunch of chains of amino acids that extend out and up and, How is glycogen different from starch? Anybody remember? Starch is a single chain of multiple glucose molecules. It's just one long chain. Whereas glycogen, so if each circle here represents a glucose molecule, starch is just a long chain. But glycogen is branched. Glycogen is used as an energy reserve. It's because we can't store ATP, we can store the glucose and use that later and run it through glycolysis in the citric acid cycle. Yeah?
Well, they're long chains of carbons?
Correct. All these circles, no, sorry. In this case, these are glucose molecules. I know I used circles to represent carbons in the previous diagram. Each of these is a glucose. Each of these is a glucose molecule. And the difference between starch and glycogen is that starch is just a single chain, whereas this is branched.
When you say branch, refer to branch versus chain. What are the variations and what's the difference between a branch and...
Let's say you just have a tree trunk.
Okay.
That's starch. But if you have a tree that has all these branches coming off to form the canopy of the tree, that's glycogen.
Okay.
How's that?
Thank you.
Now, if the cell's ATP needs are met, glycogen reserves are full, the other option is to convert that glucose 6-phosphate to lipids. There are biochemical pathways that allow that glucose 6-phosphate to be converted to lipids, and we'll address those as well.
Backing up on that, so you're saying it can go to glucose 6-phosphate or convert to lipids or can...
It will always go to glucose 6-phosphate. The first choice is that it will go through glycolysis. But if the cell's ATP needs are met, it doesn't need to go through glycolysis. It then
might go to glycogen.
It might go to glycogen.
Might go to lipids.
Once the glycogen reserves are full, and we still don't need to go through glycolysis, then it becomes lipids.
Okay,
So basically it's
it can go
1, 2, 3. If we don't need to go through 1, then it will go through 2. If we don't need 2, then it goes to 3. Yeah.
Okay.
Okay, so we have our glucose 6-phosphate. Debating how much I should get into this next little step. One of the things that happens with the help of an enzyme is that this glucose 6-phosphate here is converted into another molecule called fructose 6-phosphate. Basically what's happening is some hydrogens and oxygens have been shifted around, changing it from glucose to fructose. Fructose is still 6 carbons. You've heard of fructose, high fructose corn syrup. Fructose is just another 6-carbon carbohydrate. And so I've drawn this so that the triangle is on the other side, showing you that there is an enzyme that changes that glucose 6-phosphate into what's called an isomer. Very similar molecule. It still has the same formula, C6H12O6. It's just structurally, it's different. Has the same number of atoms and the same atoms. They're just arranged differently. The enzyme basically rearranges things.
Do you have to know that enzyme?
No. Only know the enzymes if I tell them to you. All right, so this one here is the priming phase. It is important to know that enzymes facilitate all these reactions. But unless I tell you what the enzyme is, don't worry about knowing it. So what we're doing then is we're adding another phosphate. And where does that phosphate come from, do you think? ATP. And what are we left with? ADP. And then our product here is called fructose 1,6-diphosphate. Fructose 1,6-diphosphate. One and 6 refer to the carbons where the phosphates are attached. So the number 1 carbon and the number 6 carbon. Now again, enzymes will split that fructose 1,6-diphosphate pretty much in half. So we're going from one 6-carbon molecule with 2 phosphates to 2 3-carbon molecules with 1 phosphate each. Okay? That was the cleavage phase that we called it. Split in half. And what we end up getting here is 2 molecules of something called phosphoglyceraldehyde, or PGAL, or glyceraldehyde 3-phosphate. So we've gone from 1 molecule with 6 carbons to 2 molecules of 3 carbons. I think I've mentioned this before. There is a law associated with chemistry, physics, biology, all that, or the universe really, and that's the law of conservation of matter, saying that matter can't be created or destroyed, but you can shift matter from one form to another and to one substance to another. And if we were to look at the amount of matter we started with at the beginning, we should be able to account for all that. So when we look at this PGAL, this glyceraldehyde 3-phosphate, the number of atoms between those 2 molecules here should be the same as what we saw up here with fructose 1,6-diphosphate. And that's something that you should pay attention to as we go through this process as well. Is that matter will not be, we're not going to create new matter. We're simply moving matter from one molecule to another. And in this process, ultimately the goal is to move matter from one molecule to another to generate as much ATP